Hey there, welcome to The Explainer. Today, we're going to unlock one of the most fundamental processes in all of life. We're talking about how the blueprint written in your DNA gets turned into, well, you.
So you ever stop and think, how does a simple little four-letter code inside your cells build something as unbelievably complex as a person? Well, today we're going to follow that exact journey, from the genetic blueprint all the way to the proteins that do all the heavy lifting.
And it all comes down to this one core concept, known as the central dogma of biology. It's really the master plan for pretty much all living things. Just think of it as this universal chain of command that's happening inside every single one of your cells, right this second.
All right, here's our roadmap for today. First, we're going to meet the molecular team that gets all this work done. Then we'll see how the DNA code gets copied, how that copy is decoded, and finally, how it's used to actually build a protein. And it all ties back to this really powerful idea. One gene makes one protein. Okay, let's dive in.
All right, first things first. Before we can build anything, we have to meet the specialized crew that makes it all happen. We're talking about the three main types of RNA.
So each type of RNA has a super specific job. And honestly, the easiest way to think about it is like a construction project. You've got your mRNA, or messenger RNA. That's the temporary copy of the blueprint. Then there's rRNA, ribosomal RNA, which helps build the factory itself, called the ribosome. And finally, tRNA, or transfer RNA, which acts like the delivery trucks, bringing all the raw materials, the amino acids, to the construction site.
And here's what they actually look like. It's pretty cool, right? You can see how their shape is perfectly suited for their job. The mRNA is just this long, simple strand of information, super easy to read. The tRNA has this distinctive cloverleaf shape that works like a key, carrying a specific amino acid on one end and the code to match the blueprint on the other. And the RNA? It's a huge part of the factory itself. It is the machinery.
Okay, let's pause for a quick knowledge check to make sure we've met the whole team.
All right, now that we've met the team, let's see how the cell actually makes a copy of its master blueprint. This process is called transcription.
And this whole thing is actually pretty straightforward. It happens in three clear steps. First up is initiation, where the DNA gets unzipped. Then, during elongation, this key enzyme, RNA polymerase, scoots along one strand of the DNA, building a matching mRNA copy. And finally, there's termination, where the enzyme hits a stop signal and releases the brand new mRNA copy.
And this is the star of the show, RNA polymerase. Just look at it go. It's cruising down the DNA strand, unzipping the double helix as it goes, and using one side as a template to build that perfect mRNA copy. It is literally a molecular copy machine. But you know, in more complex cells like ours, we call them eukaryotic cells, there's a little bit of an editing process. The first copy, the pre-mRNA, has these junk sections called introns that don't really code for anything. So those get snipped out. The important parts, the exons, get stitched together. And then, a little protective cap and a long tail are added to make sure the message survives its journey out of the nucleus.
Time for another check-in. How well did you follow that whole copying process?
So we've got our message. It's been copied, it's been edited, but how does the cell actually read a language that only has 4 letters, A, U, G, and C, to build proteins that are made from 20 different building blocks?
Well, the answer is kind of just math and logic. Let's think about it. If you used one letter, that's only 4 options, not nearly enough for the 20 amino acids. What about 2 letters? Well, that gives you 16 combinations. Still not enough. But 3 letters? That gives you 64 possible combinations. More than enough for the job. And that's exactly what the cell does. It reads the code in these 3-letter words, and each one is called a codon. Every codon is like a specific instruction in the recipe book, either calling for a particular amino acid or simply saying, hey, stop building now. And this right here, this is basically the universal dictionary for life. Seriously, almost every living thing on Earth uses this exact same code. To read it, you start in the middle and work your way out. So for example, A, U, G, that codes for the amino acid methionine, and that's also the universal start signal. And then you see others like UAA, which is one of the stop signals.
Okay, let's see if you're speaking the language of the cell now.
All right, now for the main event. We're taking that mRNA message and we're actually going to build something with it. This process is called translation, and it all goes down on the factory floor of the cell, at the ribosome.
This diagram is amazing because it puts everything together. You can see the finished mRNA leaving the nucleus. Out in the cytoplasm, it finds a ribosome, our factory.
Then the delivery trucks, the tRNA molecules, start showing up, each one carrying a specific amino acid. Now here's the magic. Each tRNA has a 3-base sequence called an anticodon, which is a perfect mirror image of the mRNA's codon. When they lock together, the tRNA drops off its amino acid, and the ribosome links it to the growing chain. It's an assembly line, building a protein one amino acid at a time.
Let's just check our understanding of that final assembly process.
Okay, so let's zoom out one last time and connect all these little steps back to the big, powerful idea behind it all.
And that is the one gene, one protein hypothesis. It's so simple, but so profound. It basically says that this entire incredibly complex process we've just walked through exists to do one thing: turn the information in a single gene into a single functional product like an enzyme. Now we know there are a few exceptions, but this is still a foundational concept in all of biology.
One last quiz to put it all together.
So we've seen this beautiful, perfect system, this incredible factory line working flawlessly. But what happens when there's a typo? A tiny mistake in that original DNA code? Well, a single mutation can change the message, change the protein, and have massive consequences. And that is a story for another explainer.